for your lipids. So now what we're going to do is see what we can do with those lipids. So how can we arrange them? And that's ultimately, for us, the biological membranes. And we talked about those three different types. Primarily, we're going to be concerned with the bilayer sheet, which really, as far as a cell goes, that's really what we're looking at, is that bilayer sheet. It's not really a sheet, though. It is just a really long sheet that now can wrap around. So within any small space, it's effectively flat. Right? That's why we can call it a sheet. As far as the composition of those, hydrophilic outwards, hydrophobic inwards, and then we can set up that membrane. I mean, relatively simple as far as that goes. Your plants typically are unsaturated, which we've been kind of discussing when we went through the lipids. That unsaturation makes the lipids more, or the membrane more fluid. Pretty sure I've got a nice picture that'll show that. And then in animals, it ends up being less fluid. So you're looking at more saturated fatty acids. Okay, you're getting those straighter chains. So what makes up the membranes? So we can pretty much put in any type of lipid we want. And depending on what we're trying to get out of the membrane or what we're trying to put into the membrane, that composition is going to be slightly different. Okay? So your textbook has this kind of nice picture, and they've color-coded it. So if you look at the outside of the wider section of the membrane, you're looking at a whole kind of smattering of different colors. So you've got some reds, and you've got some purples, and some greens, and all that fun stuff, right? Okay, kind of going around on the outside. But if you look to the inside, you'll notice that it's fairly consistent. We only get one type. Okay, why might that be the case? Are we going to expect to see much of a difference between hydrophilicity in that case? The outside is having charge, the inside has charge. So the hydrophilicity probably isn't that big of a difference.
Oh, or proteins taken outside of that?
It's going to be part of it. When we look at a sphere, okay, or a circle, okay, where do we have larger space? So if we take a look, the gap between these two lipid ends over here is quite large, whereas if we shift to the inside, what does that gap become? Much smaller because, yeah, it's looking at a surface area change. As soon as we get smaller, we have to then pack things in tighter. So what is that going to mean about what we would expect for our lipids on the inside? We're going to expect a smaller polar head group, because if it's larger, it's going to then actually cause it to bow outwards, and we might get a pocket. Instead of it curving like a nice sphere all the way around, maybe it'll end up curving like a big pocket, okay, to account for that larger shape. Okay, so we want to try and keep that as consistent as possible. So the inner versus the outer membrane are going to have slightly different compositions to kind of account for that. Okay. Beyond that, we could also look at the fatty acid portions of it. I don't think this is the figure that I'm looking for in this case. But if we've got saturated fatty acids, we can get the nice straight chains running all the way through. If we have unsaturated fatty acids, remember we have those cis double bonds. Okay, and it's nice of biochemists to give us this drawing. But they made those double bonds really long so they could make it nice and neat in a straight chain. Is it going to be nice and neat? No, it's going to cause a kink in it, which is going to cause the spacing to change between those amino acids, or sorry, fatty acids. The spherical shape is because the hydrophilic areas are trying to interact with water, and the hydrophobic is trying to interact away from water. So that's ultimately what's generating our spherical shape. What those kinks are going to do is now increase the spacing between that. Okay, well, what is that going to do for us?
Allow transport.
It's going to allow for easier transport of things through, because now I don't have to get by as much junk to pass into the inner portions. Okay? So by changing the fatty portion of our lipids, we're going to change ultimately the dynamics of that membrane, and it could become more porous or less porous depending on what we're working with. Okay? Membrane fluidity. So this is the figure I was looking at. Okay, if we take a look at our saturated versus our unsaturated, they did a much nicer job with this. You can see those kinks are now going to cause kind of a kick out. That's going to prevent the next fatty acid from, or the next lipid from stacking up against it quite as nicely. Okay? And that's going to then prevent that organization. Okay? So we won't get it as densely packed. So unsaturated becomes more fluid. So if you remember back to right here, within our plants, our unsaturated fatty acids generate a more fluid membrane. Okay?
So what is the definition of fluid then? 'Cause I would think
Fluid means more mobile.
fluid saturated is more mobile.
So what's happening in the saturated situation
More tight.
is I'm going to now put in a second molecule. Here's my polar head group. That fatty acid chain can now align directly next to it. If I try and do that same thing with the next one, can I get that nice, neat, tight stacking? No. So that's going to disrupt the intermolecular forces, preventing it from holding as tightly. Okay, so unsaturation will actually allow more motion or more space between them because there's less things holding it together. We get less direct surface area overlap. Okay. We can also look at a change in the amount of cholesterol showing up within it. So cholesterol was technically a lipid, though it doesn't have quite as polar a head group as the other ones. Why is it not as polar? Head group on most of our lipids was phosphate. Why is the phosphate more polar than our cholesterol head group? It's charged. Yeah, most of our lipids had a charged head group. That charge is going to be very water soluble or very hydrophilic. With our cholesterol, yes, we have a hydrophilic head group, but it's just an alcohol. Okay, it's not charged. We're only going to get kind of a relatively weaker hydrogen bonding interaction. Okay, and it's also much, much smaller in comparison to the rest of that structure. So cholesterol doesn't quite as interact quite as neatly as it would with the rest of those lipids. Okay. What is cholesterol going to do as far as the overall structure, though? So any guesses? It's bulkier, so what might you expect to happen to the interactions between those chains? Get weaker or stronger? You might expect it to get weaker. Okay. Turns out that there's actually kind of an odd effect going on there. What's happening is because of that ring structure, it's now forced into effectively a straight chain. Whereas with our standard chains, what can those sigma bonds do? They can freely rotate. So those can rotate and kick out just like our cis-trans, just not quite as frequently. So we still expect them to have stronger intermolecular forces than our cis-trans or than our unsaturated structures. But they can still kick and move and cause other fatty acids to shift away from it. In the case of cholesterol, so number one, do we have ring strain? This is a six-membered ring, six-membered ring, six-membered ring. We aren't going to have any real ring strain. What we do have, though, is those fused rings now kind of limit the ability to ring flip. So we now have all of that hydrophobic interaction, and I can't move it. Okay. If it can't possibly move, what's going to happen?
It's going to keep it stable.
It's going to maintain straight chains for our saturated fatty acids around it. If those maintain their straight chain formation, what happens to the fluidity?
It goes down.
It goes down. Okay? It becomes less fluid. So cholesterol actually prevents a fluid membrane.
So in colder environments, you expect it to have more cholesterol in your membrane?
So if we want to look at colder environments, what's going to happen ultimately when we decrease the temperature?
Solidifies.
It's going to solidify everything. So if we want to actually keep mobility within the cell, right, what are we going to expect to see within the fatty acid chains? More kinks. So that as it cools down, it can't fully stack back on itself. So it will reduce the fluidity, but it still has that ability to move. Because in this straight chain formation, when we reduce the temperature, we're now going to really drastically shrink those down, and there's now no way for it to adjust. So in colder environments, we'd expect less of that, okay, and a little bit more kinkage to it so that we can maintain that fluidity. Okay. Cholesterol is going to kind of shrink that fluidity down the other direction, and we're going to lose that fluidity. Where does cholesterol show up? Cell membranes and just in animals. You don't see it in plants.
So that's why it's bad in high cholesterol.
We can't. No, we don't want to open that can.
I was just curious.
There's all sorts of arguments against the whole high cholesterol issue. The initial findings were apparently faked.
Thanks. Really?
Yeah.
it's fake.
So there's a high correlation, but there's very little causation right now between cholesterol and heart disease. Where was I going? Oh, so no, you're fine. So in animals, we tend to get this cholesterol. Why do we not see it in plants?
Well, they're more fluid anyway.
They're more fluid membranes, but they have a cell wall. Animals don't have that cell wall. Okay, so what ends up happening is we need still something to kind of maintain that shape. That shape is going to be contributed by cholesterol. Okay, so cholesterol effectively gives animals a cell wall. Okay, whereas plants have a secondary cell wall, they don't need that extra protection. Okay?
So cholesterol is like the main that gives it shape, like
I don't know if I want to say shape necessarily.
more of a rigidity.
It's going to give it more or a more rigid structure. Okay, so it's going to prevent things from moving as much.
So our phospholipid bilayer is composed partially of cholesterol,
Our phospholipid bilayer is made up of lipids, period.
and those are cholesterol.
And so if we look at that example, you do see that we have some pockets of cholesterol. But the entire membrane or the entire bilayer is going to be made up of all our possible lipids. And depending on what we want out of that particular cell, maybe we want something relatively rigid. In that case, we'll probably see a lot more cholesterol.
So basically,
showing
those
up.
just keep the ones in between straighter.
Yep. So it'll maintain and force that geometry to increase intermolecular forces. Okay. General models of your membranes. We still need to be able to transport stuff through these membranes. So we can't just have a solid wall that prevents all passage of things. Okay, so cells have developed proteins to allow for the passage of stuff through it, or passage of stuff through the membrane, and we really need those proteins to help that transport. Okay, and that's because when we hit the surface of the membrane, it's hydrophilic. So we could see a strong attraction of maybe ions to the surface, but then how do we get those ions to pass through the membrane? They'd have to go through a hydrophobic section. We're going to see a really strong repulsive force. The ability for those ions to pass through is going to be pretty limited. So we need something to help ease that transportation process across, and that's where we're going to incorporate proteins. So proteins will kind of transition that barrier because our proteins could effectively build holes that could run through the middle of the membrane and allow for the passage of ions. Okay, so we still need that transport ability. Further, we could also look at receptor proteins. So perhaps we just want the cell to acknowledge that something else is present and change something else within the cell. Okay? So in that case, we're just going to have something on the surface. When it detects another molecule, it changes something within the cell that accounts for that, whatever that receptor was. Okay? So
Concentration gradient charges.
we could look at concentration gradients and charges and things like that, and detecting when to function along those lines. So we really have these kind of two big classes. We get your transport, which allow for the direct passage through, receptors, which will allow for changes on either side of the membrane. Okay. Further, we're going to have to decide how do we do each of those things. So a transport protein is going to have to run through the middle of the membrane so that it can allow for the passage of stuff directly through. Well, in that case, we have something that's integrated into the membrane. We now have an integral membrane, integral protein. Okay. A receptor, on the other hand, may not necessarily need to actually run all the way through the membrane. Okay. It could potentially make changes on the surface. So in those cases, what we'd be looking at is peripheral proteins.
So the peripheral ones more like the ones that change, like something just attached.
Could potentially change. So what we could be looking at a peripheral is something binding and then interacting with a transport or an integral protein to cause a change on the other side. They won't directly pass through. Yeah. So your peripheral are just on that membrane surface. Okay. Either on the, do we have an inside outside in this? Yes, we do. We are either on the outside or on the inside. Okay. How do we know any of this is actually true? Effectively true. What we want to do is, or come up with some way to actually visualize this. So one of the methods they came up with is this freeze fracture technique. So what we can do is freeze the cells, and then if you hit the cells or hit that ice properly, what can end up happening is you can then cleave the membrane directly open. Okay, and that's this updated version of your textbook actually has a much nicer picture, but I couldn't find that. So what we've got is effectively this kind of idea. We've frozen the cell, and then when we cleave it, we can actually split the membrane right down that middle, and then we can see the pockets of what's in it and what's not in it. Okay. As a semi-gross example, you could do like, what are those pore remover things, blackhead remover, stick it on and peel it. You can see little things poking up at you. That's effectively what we're doing here. Okay. You're getting kind of a negative image of that. And then we can then go through, it's not directly scanning it, but we can use a microscope to scan over the surface. And you notice that you see all these kind of little bumps. What are all the little bumps? Those are your proteins, and then it's a question of are they peripheral or not. That's a different question. To figure that out is we'd have to figure out how tall that bump was. If it's relatively short, it's probably just stuck within the membrane. If it's a really tall one, it probably pulled all the way through the membrane. So you'd have to look a little bit more carefully to figure that out. Okay? We still have to, so this is the fluid. I don't know why I put this all on the same slide, because these are two different topics. So freeze fracture allows us to break it open so that we could see what's actually happening within it. The other aspect of this is what they refer to as the fluid mosaic model for your membranes. Okay? They are constantly moving, which means your proteins are constantly moving through that membrane as well. So don't look at these membranes as a stationary process. You've got a lot of things kind of shifting around within these.
So you're saying they're just kind of like...
So you could look at it almost as a bunch of boats on the ocean.
Okay.
Okay? Those boats kind of floating through the ocean and now interacting with other things. Okay? And that's where they're getting the fluid idea from. The mosaic part is that there are proteins that need to communicate with other proteins. Okay? So we're going to maintain a relatively close proximity between those two because they're relatively partnered. So while we do have the ability to shift around, some proteins are going to be closer to others to allow for that direct transport a lot easier. Okay? And then how do we know about this proximity issue or this need for the proximity? GFP with that question mark. Anybody know what GFP stands for?
It's the protein that you.
P is protein, yes.
Fluorescent.
Green fluorescent protein. Okay.
Just did that yesterday
Oh yeah, cool.
in microbiome.
Oh, nice. So green fluorescent protein is a very common tag, and it can be sensitive to things that are around it. So what we can use is a variety of different tags, and that if those two tags get close enough to each other, we now see a particular color. Okay? We see a fluorescence. If they never get close to each other, then we know that those two things that we tagged don't ever get near each other. Okay? We could use these as well in larger enzymes or larger protein structures, because we could tag some part of the protein, and if a substrate gets close enough to it or another part of the enzyme folds around and gets near it, and we see that fluorescence, we know, oh, okay, those two things had to fold close to each other. If they don't ever get close to each other, it doesn't allow for the transport of light energy appropriately, and we don't see that fluorescence. Okay, so we can use GFP and others like it, other fluorescent proteins like it, to actually tag proximity or determine relative proximities of things ultimately anywhere within a cell. Okay, so it's kind of neat in that respect. The membrane transport control. So we're going to have two types of methods. You've got a passive transport and you have an active transport. Your passive are driven based on concentration differences. So if there's a large concentration gradient, we're going to start to see ions pass through to balance that concentration gradient. Right? That's still going to have to pass through something, okay? Because it's not super easy for it to directly pass through the membrane. So there's usually going to be some protein involved with that either is going to facilitate that transfer directly or take advantage of that transport. Okay? We've got two types of diffusion processes in that passive. You've got simple diffusion, so that just says there's a hole, and whatever you're trying to diffuse through passes directly through the hole, and there it is, okay, and it's made it across. The other option is your facilitated diffusion. You're actually getting a direct transport. Okay? So a protein is actually carrying it ultimately from one side of the membrane to the other.
Is facilitated, does it usually go against the gradient? Or
These are, your passive still go with the gradient. Yeah. So in those cases, you still have to have that protein there to help kind of shuttle it across and carry it across. In the active transport, that's where you've got it driving against the gradient. Okay? So you could look at that electron transport chain, which it sounds like you guys know more about it than I do. But in your initial electron transport, we start driving hydrogen ions across the membrane. So we build up an H+ gradient. In that case, what we're looking at is an active system, because we're actually trying to make a gradient.
and when it comes back?
And then at the very end of that, you've got ATP synthase that is allowing for the shift of hydrogen ions back across, that's allowing for the formation of ATP. Okay? So within that, we actually have two transport mechanisms. We have both a passive and an active.
Is there a reason they're leaving the sodium potassium pumps alone with concentration gradients with this? I think that would be part of this.
No, it is part of it. I just didn't put it up as a slide. And did I not put it up? No, the sodium potassium one is in the lower right-hand corner. Their figure is awful again. In the newer edition of the textbook, they have a really nice figure for it. And this one, it's not quite as nice. They actually use pictures and everything in the newer version. But you are seeing the transport of sodium and potassium ions. Okay? And we need ATP to drive that. Okay? So there's an energy source to actually force that transition of those ions across the membrane. Okay? So we get those two methods, either a primary transport or a secondary transport. Your primary transport uses HTP, ATP, and your secondary transport takes advantage of the H+ gradient. All right. The receptors. Sorry, I really don't have a whole lot to say about the receptors. They're relatively short proteins. We want them to be relatively small because they're sitting just on the surface. Remember, they're peripheral proteins. They aren't going to pass all the way through. So they're going to be relatively small. So we're looking at relatively short protein structures. And if we're looking at a short protein, that's where you get this term oligomeric. It's really just saying you have a very short polymer. That's all that means. I don't know why we had to invent a new word for that.
Basically, the peripherals.
And that's going to be your peripherals, yeah, which are ultimately going to be your receptors as well. Okay? So when we bind to that receptor, it changes something that allows for either the passage of something else or maybe just direct communication across. Saying, oh, there's a bunch of acid in here. We need to move this cell away from this location because it's going to destroy us. That kind of thing. Okay? And yeah, no fun pictures. Now we're moving in pretty much the very end of this chapter. Excuse me. You're going to look at your lipid-soluble vitamins. The textbook lists off a couple others, but I picked two that I thought were particularly interesting, vitamin A and vitamin E. Vitamin A is involved in sight. So vitamin A is derived from beta-carotene, which we can get from like carrots and such. And what ends up happening is we can cleave that open and we can get our vitamin A. Vitamin A is really important in sight because what ends up happening through a sequence of other pathways is we convert retinol or vitamin A and bind it to the receptors within your eyes. Okay? Then what happens is that we can see a cis-trans isomerization when it absorbs light energy. Okay? That change in structure is then detected and translated into sight. Okay? As a very superficial way, and I'm sure Stephanie can explain it better. Okay? But that's kind of a neat process because you're going back to the organic chemistry, looking at the absorption of light around those double bonds. And that's where you're seeing your light. So it's kind of neat. Vitamin E is an antioxidant. What do antioxidants do? Yeah, you're right. That's why I covered it up real quickly. They're free radical scavengers. So oxidation, you're looking at free radicals, usually peroxides, that kind of things. Those are really bad. So we've got oxygen radicals floating around. What's the issue with a radical? They're really reactive, and they'll react with pretty much anything they come in contact with. Okay, so they're very helpful in the respect that we now have a high energy state that we could then use. But if they're generated and we aren't using them, then they can do damage. Okay, so we want to only generate these kind of radical situations when we can actually take advantage of that energy. Okay, so because we're looking at a free-floating cell where things could be shifting all over the place, we could potentially see these radicals diffuse somewhere where we don't want them to be. Okay, so there has to be some way to kind of collect and trap those radicals. Vitamin E happens to do a really good job of doing that. Why might vitamin E scavenge free radicals? So positive and negative is generally a good concept to go through, except we aren't positive and we aren't negative. So we can't use that kind of nucleophile, electrophile definition that we would like to use from organic chemistry. And radicals show up in organic, but they're typically kind of skipped over.
They donate a hydrogen.
All that resonance
Kinda.
except the hydrogen.
There's no other, all that resonance, it's one ring.
On the oxygen.
So we get a little bit of resonance with our oxygen, but we've got lots of phenol groups. I mean, you could look at amino acids have that same functionality. Why would this be that much better? Where's our radical most stable? Yeah, where are radicals the most stable? And now I'm going to be really mean to everybody from Mesa.
When bonded to hydrogen.
Yeah, we'll do that.
Yeah. BRZ,
What's your product?
B at the end, for the CHC.
Nope.
Oh, isn't it
Come
right,
on, Brady,
right,
this
right
is
where
your
the
question.
ring meets?
Orthopara, which one is it? Is it?
It's not an orthopara situation, at least the way I've got it drawn,
It's
because it's not a benzene ring. Orthoparameta only works on benzene. This is cyclohexane. Okay? I can, if you still want to go with the orthometapara analogy, I can go through and make that a benzene ring. Doesn't change the answer.
It's the most substituted.
It has to do with the most substituted. What's going on with your radicals? We need to form the most stable radical possible. Where's our most stable radical? Or why are radicals unstable?
The most substituted from.
They're missing electrons.
So you want to be on.
We want to be on the most substituted position possible. Okay. Why might vitamin E work out really nicely? We've got a lot of tertiary hydrogens, which could then be substituted out and generate our radicals. So vitamin E can react relatively quickly and easily with our free radicals to kind of hold those and stabilize them until we can find a better way to alleviate that stress. Whereas our oxygen free radicals are just going to do whatever the heck they want. Okay? They're going to go out really fast. So vitamin E allows us to take that very highly reactive oxygen radical and temporarily stabilize it and hopefully provide the cell enough time to actually dissipate that radical energy in a more productive fashion.
We can just pass out.
It doesn't get rid of the radical tube. Okay, remember, what's going to happen is you'll pull off a hydrogen at any one of those kind of tertiary sites. Okay, but now you have that radical. Why is that better than the oxygen? It's going to be lower energy because it's a tertiary radical versus really a primary for oxygen. Okay. I think they talk about vitamin K, vitamin B maybe. I don't remember exactly what other vitamins they talked about. But all they're really doing is saying this is what they do. Okay, just like we went through here. This is what they do. Okay, so you should read through that just to get an idea of what's going on with it. I just picked the two that I thought were coolest. The very end of this, we can look at arachidonic acid derivatives. So arachidonic acid is that structure shown in the upper right-hand corner. For some reason, I want to say 20 carbons, but I'm not going to count that. So we've got that general structure, and you notice that we have a bunch of double bonds in there. Okay. What could we do potentially with a bunch of double bonds?
20 carbons.
Was it 20 carbons? Yeah, sweet. Sometimes my memory works.
You can make them more substitute.
What can we do with a bunch of double carbons?
Double bonds.
Break him.
Release
We
energy.
could break them and put other structures in. Take a guess based on what you see down in here.
Add hydrogen.
What's happening in these cases? We're doing sigmatropic reactions, cyclization reactions. Okay. So we can actually make rings by having those double bonds close enough to each other. Okay. And that's what ends up happening with some of these. So your prostaglandins, originally found in the prostate, hence the name, generate a whole bunch of different relatively hydrophobic species. And you're looking at these kind of derivatives with those general 5-membered rings and those long carbon chains coming off of them. They have all sorts of different chemical usages, which is allegedly why drug manufacturers are really interested in them, because you get smooth muscle contractions, blood pressure, induction of inflammation. So you've got all of those possible kind of things that you could play around with if you can make derivatives that match these. Okay? And then you get your leukotrienes. These are typically found in white blood cells and located primarily for the constriction of smooth muscles. You'll notice again it's very, very similar to your arachidonic acid structure. The only thing that really changed is we switched out one of those double bonds for that sulfur group. Actually, we didn't even do that because there's still 4 double bonds in that structure. Yeah. Which is kind of fun. What does tri mean? 3 and ene. Double bonds. So the reason I'm kind of hesitant here on discussing this is I've definitely noticed the textbook has drawn some organic chemistry kind of things really badly, and this might be a typo. If they're going to refer to that as a triene, and they're showing 4 double bonds, I'm not sure what exactly they're trying to do. So just be kind of aware there might be a typo in that situation, and this was definitely pulled from the textbook. What they're primarily concerned about is what those leukotrienes will do. You're going into the white blood cells and you're looking at smooth muscle constriction. Okay? And again, a derivative of the arachidonic acid.
Maybe those 3 double bonds are
Yeah, they could just be referring to those 3. But I don't know.
That's what I was wondering too.
Yeah,
Those are part of the ring.
Yeah.
Or that part.
Well, those would probably be involved in the prostaglandins, so I'm trying to drop down into that 5-membered ring. Beyond that, it's kind of hard to say. Yeah. Okay. So that's pretty much all of our lipids. Are you guys Google searching leukotrienes by any chance? Yeah. Does it show 4 double bonds?
Yeah,
It does. Okay.
Not yet. Okay.
Oh, that's right. You guys are looking for pharmacy applications.
Today might be the day.
Yeah. So chapter 16 moves into the carbohydrates. So probably what we'll get through today is just kind of the general primary structure. I do apologize. You'll notice that these slides look nothing like the way I typically set up the slides, and that's because they're just the textbook slides. I did some tweaks, and hopefully everything that we'll get through today would be the tweaks that I made to it, and we won't get into any of the raw textbook slides. I just don't find them quite as interesting. So our carbohydrates are ultimately polymers. So we're looking at a monomer, or one monomer within a carbohydrate is a monosaccharide. Okay? And that's ultimately just the definition that it's a carbohydrate that can't be simplified further down into other carbohydrates. Okay? It's kind of an odd definition because, at least the way I've organized it, we're referring to monosaccharides as a carbohydrate without defining carbohydrates. So carbohydrates are polyhydroxyaldehydes or ketones that could be potentially linked with each other. And that's kind of ultimately it. Our general formula for these is we have that CnH2nOn, so we have the same number of oxygens as we do carbons. And then as far as what we're concerned about with our carbohydrates, we're varying between 3 to 8 carbons. Why would we stick in that 3 to 8 range and not go more or less? Okay, let's try the more end of it. Why not go more?
Solubility, right?
If we've got all those oxygens on there, I would make the argument that we're not going to have a solubility issue. Okay, if it was just one oxygen, I'd say yeah, but we've got the exact same number of oxygens as we have carbons, so that shouldn't be a solubility issue. It probably has to do with the ring structure, and cells have ultimately decided that anything more than 8 carbons they just couldn't use. Okay, so we're trying to condense those into kind of the most stable situation that we can manipulate and use later on. Okay, so beyond 8 is probably just a little bit too big for the cell to actually manage appropriately. Why not go less than, or why not go less than, number less than 3? Why do we not have 2 or 1 carbons? What's our definition of our carbohydrates? Polyhydroxyaldehyde. What does poly mean? More than one hydroxy and an aldehyde. If we had 2 carbons, carbon, carbon, here's an aldehyde, how do we make it polyhydroxy? Okay, we'd have to really add another carbon, okay, as far as this is concerned. Otherwise, we start exceeding our oxygen to carbon ratio. So we're really sticking in that 3 to 8 range for our carbohydrates.
Aldehyde is it normally 2 hydrogens on the carbon?
No, it's 1 hydrogen.
It's 1 hydrogen.
If we shift to what you're thinking, I believe, is this guy. Yeah, is formaldehyde. So it is still an aldehyde, but it's a very special case. So that's formaldehyde. Sorry, were you guys drawing that?
Just trying to get
Draw that back. So we had formaldehyde. I kind of ran out of space there. And then with 2 carbons, we can put on that aldehyde, but then we kind of lose the ratio that makes up our definition, okay, if we tried to put on another oxygen. So we can't quite get a polyhydroxyaldehyde with 2 carbons. So we need at least 3 carbons. Okay. These can get subcategorized into 2 different situations. You can get aldoses and ketoses. What are we referring to in those cases?
Or the location of the carboxyl.
Don't call it a carboxyl.
Or the carbonyl.
Where's the location of the carbonyl in an aldose?
At the end.
At the end. So instead of referring to it as a carbonyl, what could we say we've got as a functional group? An aldehyde. Ald for aldehyde. In the ketose, where's our carbonyl?
Located within the chain.
Okay, within our chain, which means what functional group do we have?
Ketone.
We have a ketone, hence the ket of our ketose. Okay, so we've got a couple classifications now within this, aldoses and ketoses. And we're again sitting in that 3 to 8 range for the number of carbons. Okay? Questions on those kind of definitions? Okay, because then it turns into a mess, in my opinion. So we'll start with our trioses, so we'll go with the monosaccharides. I hope to get some time to actually draw these in, but I didn't. So we're looking at our triose. So we know we're looking at 3 carbons in each of these. So what do you think? Carbonyl is our primary functional group, and we can scatter our hydroxys around it. Where should the carbonyl be in glyceraldehyde? As an aldehyde. So it's got to be at the end of our structure. I'm going to put it up at the top, and we'll talk about why in a little bit. How about the dihydroxyacetone? going to have to be in the middle because we have a ketone, the ONE ending. Okay, we can then go through and put on our hydroxys. In the glyceraldehyde, it's ultimately not going to matter where I put the hydroxys. How about in the dihydroxyacetone, it's not going to matter where I put the hydroxys. How about in the glyceraldehyde? Will it matter here?
It could.
Could's a good answer. Nice, safe answer. I like that.
I do too.
Is there a difference between these two structures? What's that difference? We have different chiral centers. We have a chiral center in that central carbon. All right, so one of the things that's going to be a bit tedious when we look at our carbohydrates is that we do have chiral centers. Okay? A chiral center is a carbon, as far as we're concerned, with 4 distinguishable groups attached. So if we take a look at our top situation, we have a carbon versus an oxygen versus a hydrogen versus a carbon. Is that carbon different from carbon number 1?
Yes,
Why?
because of the double bond and the oxygen.
We're looking at an aldehyde carbon versus an alcohol carbon. So those are definitely distinguishable groups. So we have 4 different things attached to that carbon. We do have a chiral center at that carbon, which means where I put that OH can change its structure. If we go through and take a look at the dihydroxyketone, why does that not become a problem? We don't have a carbon with 4 distinguishable groups attached. Okay. For sure, we could go through and label our carbon and our oxygen and one of the hydrogens, but we can't tell a difference now between those 2. So we only have 3 groups. So we don't have to worry about multiple structures with our dihydroxyketone. We do have to worry about it for our glyceraldehyde. If you remember way back at the beginning of the semester, we did talk about glyceraldehyde. What was that discussion with our glyceraldehyde? I know, it was a long time ago.
Yes.
He was ahead.
The only hit I can come up with is the answer. Just fine, I don't have a problem with that.
I already knew.
Where's our D and L coming from?
The chiral.
Our capital D and L is in reference back to glyceraldehyde. So when we look at our carbohydrates and we say, oh, it's a D carbohydrate versus an L carbohydrate, it's because that second to last carbon has the OH group, or our higher priority group, on the right-hand side of the structure, dextro for D, or on the left-hand side of the structure, levo for, or L for levo, one way or the other.
Left.
Okay, at the very least it's left. Let's stick with that.
L for left.
Okay? So all of our sugars are going to get this classification that comes back to that specific carbon.
So it's like L-arginine kind of thing.
If you look at L-arginine, the reason we call it L-arginine is because when we organize it and look at the chiral center, our more important group is on the left-hand side. Okay. To do this classification, we must be very careful with how we've got this drawn. Okay. For instance, if I redraw that L structure, so let me erase the dihydroxyacetone, or cross that out. So I'm going to redraw the L structure. Here's our carbon, carbon, carbon. Put the aldehyde down here.
Oh, that's fine.
When I do this flip,
Yeah, the highest priority at the top, right? The aldehyde.
what's the result? If I again take a look at that second to last carbon, that looks like the highest priority is on the right. Shouldn't that be a D? But it's the exact same structure, so it can't possibly change.
Just go.
What's the issue?
You have to
Is it rigid?
Yeah, the aldehyde at the top.
aldehyde.
We have to make sure that when we're looking at our structures, our carbohydrates, that we are looking at them in a very particular fashion. That very particular fashion is known as the Fischer projection. We have to be in the Fischer projection. If you are not looking at the structure within the Fischer projection, you cannot assign D and L without doing some mental manipulations. Okay? So our Fischer projection rules, we did talk about these already, but we're looking at our highest priority carbon at the top of our structure, and then our carbon chain will fall directly underneath it, okay, on that vertical axis. If that was it, we could potentially organize our carbon chain, I wish I brought a model kit, where that we had our highest priority groups coming up at us as we went through this structure. In that orientation, that's technically not the Fischer projection. Okay? So it's not just that our carbon chain falls on the vertical axis. It's that each of those carbons must be behind the previous carbon. Okay? It has to be organized such that it is away.
I was thinking it was the arms are going to hug you.
We'll come to that analysis in a second, but yeah, you're right.
That's what we learned. It was grandma coming down.
Yeah, exactly.
Grandma coming to hug you?
Or whoever. Whoever you want.
Definitely had a strange lady come up and hug me this weekend. It was very weird.
I'm going to hire one.
Oh, hi, I love you so much. How are you doing? Oh, no idea who you are, woman. I think her daughter came around and said, we're over here. Okay, I understand what's going on now. So your carbon groups are going to be aimed away from you, which then means the two remaining bonds must be aimed at you. And to show that we've got that wedge situation. Okay. So the way I was told it was instead of grandma coming out to hug you, you're looking at a bow tie. Okay. So either way works as far as this analysis goes. Okay. Depending on your grandma, you may prefer the bow tie. My grandma's very nice. I wouldn't mind if she came and hugged me. So we look at the left and right. Those bonds are coming out at you. Okay? Then we can go through and assign the D and L. If it's on the right-hand side of the structure, it's D. If it's on the left-hand side of the structure, it's L. And we're looking at capital D and capital L. Why not lowercase d and lowercase l? Those lowercase d and lowercase l refer to optical activity, which may or may not match capital D and capital L. Why not plus and minus? Plus and minus is also an optical activity thing, which, again, may or may not match capital D and capital L. Why not R and S? R and S is actually different from optical activity. Okay? And it may or may not match this D or L assignment. Okay? So you have to be kind of careful when using each of those prescriptions. What we're going to be primarily concerned about is this D and L situation, which is looking at that second to last carbon and its stereochemistry. Okay? Officially, it is the penultimate carbon, so second to last. The way I would look at it is it's the last stereocenter. Okay? And I think the last stereocenter will cover you for that. Penultimate sounds too funky for me to understand. So you'll notice in that previous drawing that I had on the other slides, I didn't show all the wedges and the dashes. Okay, why? Because as long as we know we're orienting our structure in this fashion, I don't have to use the wedges and dashes. So they become implied. So if we looked at this structure, that can get simplified down to just being simple lines coming off of it. And as long as you know that that's what you're looking at, that you are indeed looking at a Fischer projection, that is absolutely valid to deal with. When you're dealing with the textbook, they're going to draw them out just as those solid lines because they are assumed to be in the Fischer projection. Okay. I haven't looked at the homework yet. I'm going to be doing that later today. I would venture a guess they're probably going to be maintaining everything within that Fischer projection as well. Okay. You should be able to convert any structure over into a Fischer projection. Okay. Questions on that? Okay. So we can move out of our Fischer projections and back into our monosaccharides. We can look at aldotetroses. So what does an aldotetros mean? So I like the tet for 4, good call. So our tetrose, we've got 4 carbons. What else can we pull out of that name? The aldo means we have an aldehyde. So we're looking at 4 carbons with an aldehyde. Our aldehyde, since we're using our Fischer projections, have to be at that very first carbon, so the carbon on top. Okay. I forgot to mention this situation with our glyceraldehyde. With our glyceraldehyde, what was the relationship between the 2 structures we drew? They were mirror images of each other. They were non-superimposable mirror images, which meant the relationship was that they were enantiomers. So as soon as you have a chiral center, you have the potential to have an enantiomer. Okay? What happens when we add an extra carbon is we're now also adding another stereocenter. As soon as we add a second stereocenter, we now increase the possible orientations that we could get. And what we end up with now are also diastereomers. Okay? In the case of a diastereomer, we again have non-superimposable structures, but those structures are not mirror images of each other. Okay? So what they've got drawn up here are the 4 possible orientations for your aldotetroses. Did they do all 4? No, they didn't.
No, then I like 3. No, 2.
They did 3.
Yeah.
Yeah. So they didn't show, was it L Triose? Yeah. So there is one more possible orientation. You could have had L Triose. So your D versus your L is all going to be dependent where?
L3 is on page 454.
Looks like it's all dependent on the 3rd carbon.
That's where I want to be careful with our terminology.
No. 3rd.
All dependent. I mean, you're technically right. It is technically the 3rd carbon. Okay. It's the second to last, our penultimate, or our last stereocenter. So if we take a look at each of these sugars, okay, and our top 2 are D-triose, or our D-erythrose and our D-triose. You notice the OH is on the right-hand side, D. If we go down and take a look at our erythrose, well, so much for that. Our OH for the D is on the right-hand side, so it's the D-erythrose. If we move to its mirror image, where's that OH? On the left-hand side, so we refer to that now as L-erythrose. Where did they come up with erythrose versus triose? Okay. I ultimately have no freaking clue. It would be nice if they had a consistent nomenclature system. They probably saw this as you're referring to erythrocytes. Okay. That might have been where they first discovered it. What does euryth mean, though? Anybody know? I don't. I'm just curious. Okay. How about 3? What does 3 sound like? To me, that sounds like 3 carbons. Are there 3 carbons? No. Okay. Freaking biochemistry nomenclature. Okay. Can't say I'm a fan of their nomenclature system. They pretty much invented a new name to refer to each of these possible situations. Lucky you. Okay. So be aware that you don't have a nice consistent way of naming the secondary part for the most part.
Are they basing it off?
This is a D structure, okay? And what we can do is refer to it as a D aldotetrose. Okay? Beyond that, whether it's erythrose or triose, is then going to be dependent on the orientation of the OHs at the other position, at that new chiral center. Okay? If that orientation is changed, then they go through and change the suffix name. Erythro means red. Mm-hmm. Well, there's red in the drawing, so of course. Okay.
Are we going to be responsible for that?
No.
Just wanted to memorize.
Yeah, I know. It's probably something that you will continue to see throughout your careers. I would guess eventually it's something that you're going to be responsible for. Okay. But I won't make you responsible for it here to a certain extent. As we continue to add carbons, we can continue to go through and change the name. So here's where that name issue showed up. With 5 carbons, we've got a pentose. Could be an aldopentose or a ketopentose, depending on if it's an aldehyde or a ketone. Okay? We also have 6 carbons, which we'll refer to as hexoses. So at least that part of the name makes sense. Hex for 6, pent for 5, tet for 4, Okay, that translates pretty nicely. But if we go through and take a look at the root names for each of these sugars, you'll notice it's
Weird.
not very intuitive.
Some of them only have like one carbon, some of them have two.
So this is what this is trying to show is all of the possible orientations. If we only have one carbon, our root name is glycero. If we have two carbons, we've got erythro and threo, depending on the orientation of that stereocenter. Okay. With the glyceraldehyde, remember we had two possibilities, but one was the enantiomer, which means we'll still use the root, glycero. When we moved to the erythro, there were two conformations for the erythro. There's the D and the L. They still have the same orientation of that second OH, which means they maintain the erythro name. Okay. Threo, same deal. You're going to have a D and an L. The only thing that will be different would be that second to last carbon, the orientation of that OH. What happens when you move up to 4 carbons? You get arabino, what is that, lyxo?
So it just gets really confusing.
Yeah, ultimately. Some of these you should be really you should actually remember. Any ideas which ones you should remember?
Probably glycerol.
Really, arabinos? I didn't know that one. You should know arabinos? Why?
Glyceraldehyde.
Because part of it's one of the proteins.
Arabinose is
They're
important.
made
in
of proteins.
biochemistry.
So glycero is going to be important. Since you guys are telling me arabino, I'll accept that. Arabino is important.
Urethra, trio.
I'd probably go with the ribo for ribose. Where's the last one that I would say is important?
Galactose.
Think of your sugars.
Right,
Glucose.
Alright.
Okay. Yeah, you can't see it. It is on there.
Right at the bottom. There you go.
There are.
Can't see it.
Oh, frick. Shouldn't have touched it. Hey, there we go. You will see a lot of these show up in different places. Like galactose shows up pretty commonly. I've seen mannose show up quite frequently. I've heard, I'm not even sure how to say that, golos, but just heard, that's about it. And I haven't, I've heard of xylose. Xylose has shown up in a few cases. Okay? And we will talk about those, but I'm not going to expect you to know those. I do expect you to know these 4. I was going to give you 3, but you know,
Well, you don't have
way to
do
volunteer.
arabinose.
You can do the 4th with the arabinos. Okay? So I personally think those first 3 are going to be important. So I would put a lot more emphasis in memorizing those 3. Okay?
The 3 on the left.
The 3 on the left. The 3 circled ones. Your glycero, your ribo, and your gluco. Okay. The glycero really only because that's the origin of D and L. That's going to be the main reason behind that. The ribo and the gluco are more complex structures. You just kind of need to pick those ones up. Okay. Trying to. No, you're going to have to turn off bulk lights.
And we're gonna get
That works.
through.
Okay, I can still see. My iPad glows, so I'm fine with that.
I was totally feeling that.
Okay. These slides are posted, so you do have these. This is not a textbook slide, though. Okay, I think I got this from, I think it was actually from Wikipedia. Okay, so you can take a look for, we'll look up your carbohydrates in Wikipedia and you might find it there. I don't remember exactly how I got to it. You'll notice that they aren't specifying the X and the Y. Why not? Because that's only going to change the rest of the name. Okay. So your ribose is concerned about that central portion. Okay. We could look at the Aldose family tree. So this is kind of an interesting thing. Again, another kind of picture stolen from the internet. We start with with glyceraldehyde up at the top. And then what we're going to end up moving to, which glyceraldehyde is this?
Dextro. D.
Fancy, yes, dextro, or our D-glyceraldehyde, because our OH is on the right-hand side. Okay. We can then add an extra carbon, and you'll notice that we get now 2 possibilities out of that So what we're talking about is changing the orientation of that extra group. That extra group is the one added towards the very top. So when we go out to 2A and 2B, what we're talking about is adding that extra stereocenter. With these two, exactly right. What we're actually looking at is not just the aldose family tree, but the D-aldose family tree. We aren't concerned about the L. What would the L look like? The exact same thing as this, except the OHs are all flopped on the other. Or that, the second to last OH is on the left-hand side. Okay. Once we've got our 2A and 2B, we can add yet another stereocenter. In the process of adding that other stereocenter, you'll notice that we can, again, kind of set up these separate branches as we move down. And so what it's trying to mimic is holding everything above it constant. So if we take a look at within that left-hand branch, the last two OHs are on the same side in both those cases. If we shift over to the right branch, our OHs are diagonal.
They're just manipulating the next one out.
So they're really just adding that extra stereocenter and manipulating it from the root above. Okay. I did show green there. Green was a horrible color to use. Let's try purple so you can see your diagonal on the other branch. Okay. Then you can add yet another stereocenter, okay, and you can set up a whole new sequence of possibilities. So every time we add a new carbon, we end up adding a lot more possible structures, okay, or arrangements. Also known as, if we look really carefully at this patterning, we could see 2 to the n. Where do you guys remember that from? It's the number of stereoisomers.
Oh, yeah.
With one chiral atom, we have one stereoisomer. Or two stereoisomers, I count, okay, because we have the mirror image. When we have 2 stereocenters, it's now 2 to the n, which gives us 4. Why do we not see 4 structures? Remember, we left the L out, so there's these 2 plus the other 2 from our L. When we move up to 3 stereocenters, 2 to the third is 8 possible structures. 1, 2, 3, 4, plus the mirror images for all of those 4.
So what's the big takeaway?
So the takeaway is seeing the relationship between these. How are we actually connecting between these? So what I wanted you to see is kind of how to build these and how to kind of organize them in some fashion that makes sense. From what I saw in the textbook, they didn't give a nice table that summarized everything. Ideally, what I would have had was ultimately this table and this table in one figure. And at least the slides didn't have it. Does the textbook actually give a nicer figure? Yeah, textbook does have a nicer figure. For some reason, they decided it wasn't important enough to talk about in the lecture slides. Okay. So what page did you say was?
455.
So page 455 in your textbook actually puts all of this together, just like I went through and did here, so that you can actually see those relationships. Okay. Otherwise, you're just putting in a bunch of OHs. Okay. Actually, I want to go back on this slide. Shoot. Maybe I don't. I don't have a clean picture for this. What we've got is an aldehyde or a ketone. What's special about the aldehyde and ketone functional group?
It can make a ring.
Indirectly. How are they reactive?
Slightly acidic.
Nope.
They have free electron pairs, and they're on a carbonyl carbon.
What's special about that carbonyl carbon?
It can be
Close to attack.
electrophile.
It is electrophilic. Okay. If we look at the rest of the sugar structure, it's made up of an aldehyde or ketone and a bunch of alcohols. What's special about our alcohols?
They're nucleophilic.
That oxygen has a bunch of lone pair density. It is actually a really good nucleophile. So what can happen is that our alcohol can attack our electrophilic carbonyl, and what we end up generating, okay, after a few mechanistic steps, is the functional group known as a hemiacetal. Good thing I filled up all of my space at the bottom for drawing. I'm going to have to erase all that.
Hang on.
Okay, fine. I'll draw it again.
I had like half the.
So we've got our carbonyl. We have our alcohol. The alcohol can attack the carbonyl. And what we'll end up with as a product, again, after a sequence of steps, is that structure, which is a hemiacetal structure. Why is it a hemiacetal? It's halfway on the way to becoming an acetal. What's an acetal? Instead of being an OH, it's an OR. Okay. So that chemistry, which in theory everybody had in organic chemistry, everybody remember that? Okay. Your hemiacetal chemistry is really, really important to carbohydrates because we've got both of those functional groups within the same structure. So what ends up happening, we can get an intramolecular hemiacetal reaction, and we end up forming a cyclic structure. So what we end up getting out of this is that cyclization between your ketals and your alcohols and your aldehydes and your alcohols. Okay. Which alcohol reacts, though? Go ahead.
The second to last carbon, the one that's the hydrocarbon.
So most typically we'll see that second to last carbon, but is it always going to be the second to last? Not necessarily. Why is it not any of the others?
They're too close.
It's too close. If you look at the ring formation in those other situations, is you end up with a ring less than or 4 or less carbons. Well, a ring of 4 or less carbons is extremely ring strained, and that ring wants to pop open. Okay, remember, these things are happening in a very fluid and mobile environment. What we're going to try and form is the most stable ring that we possibly can. So in that case, what we're going to be striving for would either be a cyclopentane or a cyclohexane. Okay? So when we're looking at our Fischer projections, you have to kind of realize that we can actually close that ring by having that alcohol react back.
So can that be either the ketone or the aldehyde?
This can be our ketone or our aldehyde.
Okay.
Okay? And depending on which it is, we're going to get slightly different results because of that.
So there's not really like you were saying, it's not always the second to last carbon.
It's not always the second to last. Yeah.
So how do you predict that? You
Ring structures. So you'd look for that ring strain in some cases, and in other cases you just kind of say, okay, which one's going to facilitate that best? It's usually the second to last. It's either the second to last or the last, I believe, are your two primary options. An issue with this, okay, or a couple issues with this reaction. Whoops. For sure, this carbon started as a chiral center. Okay, and we didn't do any chemistry to that carbon, so it's still a chiral center. Okay, so nothing changed there. But what about our carbonyl carbon? Is it a chiral center to start with? No, we only have 3. It won't go away. 3 groups around it, which means we don't have that chiral atom. What happens to that carbon in the product? It is now a chiral center. Which chiral orientation is favored? D and L were based on that second to last carbon's arrangement around the chain.
will it invert?
This is a different situation. This is a different carbon.
I don't think you can either.
Depends on where the next alcohol's at, so it's at least.
There's no stereo.
There's no stereochemical concern, at least with the structure itself. Where that concern is going to come about is what enzyme is forcing that bond to actually form. Okay? So we have 2 possible orientations for that chiral center. Okay? What are those 2 possible orientations referred to as?
RNS, yeah.
No.
R alpha and beta. Okay. I didn't invent it.
Oh man.
Why do we get 2 possible chiral centers? The alcohol can attack our carbonyl carbon p orbital from the top, the one coming out at you, or it can attack that p orbital from behind. So we end up generating two possible products, or two possible chiral centers at that point.
Which is which?
And we will refer to those as our alpha and beta. We'll talk about that in a
Is
little
there another name for racemic mixture? Like,
bit. Not that I'm aware of.
okay.
You can refer to it as by other features, like it's optically inactive. And we could refer to that reacting on its own, that potentially being a racemic point. Would we get racemic products? No, why not? Because we have chiral centers everywhere else that didn't change. So the product or the relationship between our alpha and beta, they're actually diastereomers. Okay. So we have to be aware of that position. That position is referred to as your anomeric carbon. Okay, so if we're going to look at the alpha product versus our beta product, those are known as our anomers. Okay.
Oh, that
Our-
carbon bottomed out.
Yeah. And that's going to be where we define this alpha versus beta situation. Okay. It would be nice if we just went through and referred to it as R and S. Okay. Thank you, biochemists. And instead, they come up with this alpha-beta system, which I think you could argue is going to be a little bit easier than it sounds. Oh, here's the picture I was looking at. So here's the actual drawings of everything coming together. We can have our alcohol attack our aldehyde, and we end up with our hemiacetal. So we got the general reaction form. And then we can also look at our D-glucose folding back on itself. So you can see you've got all of those other stereocenters that don't change in the process of doing this reaction. And what happens is our alcohol can either attack from the top, and if it attacks from the top, where's the new OH going to be? It's going to have to go downwards. So if our,
Where does it pick up the H
from the rest of the solution,
from? Oh, okay.
yeah, probably from the enzyme forcing it. So if we come in from the top, what ends up happening is our OH has to form downwards. So it has to go away from the attacking alcohol. What happens if our OH attacks from, I can't draw this very well, but let's say that's from the bottom. Now our OH is going to have to swing upwards, and we end up with that upward situation.
That makes sense, bottom and beta.
Whatever works.
Back.
I can accept that.
I was just trying to find a way to remember it.
Yeah, I think that might actually work. The standard way to actually relate the alpha versus the beta, what are we looking at?
So the position of the OH.
Our alpha has our OH with respect to this group,
Oh, okay.
trans. Our beta has the relationship
Cis.
cis. Okay.
Well, that's their
That is the explanation given by the textbook. I think I found counterpoints to that. But at the same time, every other site that I've looked at that tries to explain what's happening with this chemistry says cis and trans as well. Okay. So I think the reason is that in the cases that you will primarily see, you have an obvious cis-trans situation.
All right.
Okay. If the new OH is trans to your very last carbon in the carbon chain, that's your CH2OH over here. Okay. If they're trans, you call it alpha. If they're cis, you call it beta. Okay. And we're definitely out of time there, so we'll end that. We could also look at the Fischer projections for those. Thankfully, what we talked about was